Welcome to the YouTube live classes for class 12 Botany. Now we have discussed earlier regarding the Mendelian theory of inheritance, then the non-Mendelian theories like multiple alleles, then this codominance, polygenic inheritance. After that we have come up to this chromosomal theory of inheritance. Today we will discuss, under this topic we will discuss the structure of chromosome. So regarding the structure of chromosome, now we already have discussed a bit that chromosome is nothing but a combination, or this is a complex of protein and DNA molecule, and this forms the chromatin fiber which ultimately forms the chromosome. Now the chromosomal theory of inheritance is the physical basis of this chromosomal, the inheritance of chromosome, how it get inherited from one generation to another generation. So we have discussed this chromosomal theory of inheritance. So we have discussed the chromosomal theory of inheritance. The salient features, and today we will discuss regarding the chromosomes and its structure. So today's topic is structure of So this is the topic today. This is the structure of chromosome. So So regarding this we already have discussed, this is just a precap, okay. Now the chromosome are thread like structures that is located inside nucleus. Where we can found in this chromosomes? It is inside nucleus and particularly in eukaryotic cells, whereas in prokaryotic cells there is no nucleus and the chromosome is not that compact or complete structure, or it is not that organized structure as it is seen in case of eukaryotes. Now each chromosome is made up of protein and a single molecule of DNA, and it forms a compact, and it is being passed from one generation to another generation. Now the term chromosome comes from the Greek word that is chroma means color and zome means body. So it is a coloring body, it takes a particular kind of stain or nuclear stain and it comes visible when there is mitotic or meiotic phase or there is dividing phase. So these are coloring bodies, as you can see the picture here. So it takes this kind of stains, various stains are there by which we can localize chromosomes, or we can recognize, we can fix the chromosomes. So this is all about that we have discussed. Okay. Now the historical information, the term chromosome was discovered by Walter Fleming in 1882 and the term was coined by Waldier in 1888. Now let us come to the structure of chromosome. Now what is chromosome? If we will consider chromosome, now it is a complex of a single DNA molecule and a complex of proteins which are known as histone proteins. Okay. So here the proteins that are associated with the DNA molecule, they are called as histone proteins. Now we have various kind of histone proteins that we have already discussed. So this is just a quick recap. Now the histone proteins being combined with the DNA, now it forms a kind of complex molecule that is called as chromatin fiber, and this chromatin fiber in complete sense it makes a chromosome. Now the chromatin contains the vast majority of DNA of an organism, and the eukaryotic cell possesses multiple large linear chromosomes contained inside the nucleus of a cell, and the prokaryotic chromosomes. Now regarding eukaryotic chromosomes, as I have shown you here, now you can see the picture here. Now this is an eukaryotic picture where you can see lots of or numerous numbers of chromosomes are present in a diploid organism and or in a diploid cell, and they are all linear. Linear in the sense, now you can see the picture like this. Now these are all linear in structure. But in case of prokaryotes, now here the chromosomes are linear in case of eukaryotic cells, but in case of prokaryotes, now the prokaryotes, they have a single circular DNA. DNA being combined with non-histone proteins. So here the histone proteins are not there, so there is a combination of this DNA and non-histone proteins which forms the chromosome, and obviously these are not linear, rather this is a circular chromosome or circular DNA, and this forms a prokaryotic, not exactly chromosome, but this is a DNA. Now regarding the structure, now when we will come to the structure of chromosome, generally we consider the structure of eukaryotic chromosome, because chromosome is a concept of eukaryotic cell. So regarding the structure, these are the points, we will go to the picture or the diagram. Now there is a centromere, there are two chromatids at a metaphase stage, as you can see the heading here, that this is the structure of a typical metaphase chromosome. Now the centromere is there and the centromere holds two chromatids. So a single fully metaphasic chromosome has two chromatids, and there are telomeres, I will show you, and then maybe a short arm and a long arm. Okay. Now this is the complete structure of a chromosome. Now this is the region of centromere. This one is your centromere. Well, now this blue lines, this blue dotted lines or these lines, they are what? These are chromatids, okay. So these are all chromatids. So here you can well see that there are two numbers of chromatids, and they are called as what? Sister chromatids. So this point is chromatid. Or chromatids. These are two, so that is why chromatids are chromatid. Now this is the short arm, that means if this is the centromere, so from this to this is the short arm, and from this to this is the what? Long arm, as we have discussed. And finally this lower end portions, it may be like this, at this point, at this point and at this point. 4 points I can spot it here. So these are all telomere. So let us have a look. Now what are those? Centromere, chromatids, telomere, short arm and long arm. Now here you can see this is the centromere, these are the two chromatids, these are the telomeric ends, and this is the short arm, this is the long arm. Now got it? Now next, what is centromere? Now centromere is the central portion, or this is a location or a position at which the two sister chromatids they meet with each other. Well, now you can see this is a specialized DNA sequence of a chromosome that links a pair of sister chromatids. Well, now during mitosis, the spindle fibers they attach to the centromere via kinetochore. Okay, kinetochore is the region of nucleolus organizer, or here the nucleolus reorganizes after as you know, at prophase the nucleolus disorganizes and at telophase it reappears or it organizes. So, the nucleolar organizer region is the kinetochore. Okay. Now, centromeres were first thought to be genetic loci that direct the behavior of chromosome, but actually they are not exactly. So, now next comes to chromatin. Now, we have already discussed now what a chromatin is. This is a complex of protein molecules, lots of protein molecules combined with or interacting with a single DNA molecule, and it forms a kind of complex, and they are present within the nucleus of the eukaryotic cell. So, what a chromatin is? Chromatin is a complex of DNA and proteins that form chromosomes within the nucleus of eukaryotic cells. Now, these are the functions that we already have discussed. Now, the primary function is to keep or to pack the long DNA. Now, it will not be entangled or it will not be intertwined, so that when transcription occurs there will be no hindrance to this kind of DNA molecule. So, this chromatin fiber, they actually guard the DNA, they protect the DNA, and at the time of requirement they also release the DNA in order to go for transcription. Now, it prevents DNA damage also, and it regulates gene expression and DNA replication, and facilitates proper segregation of chromosomes during cell division. So, these are the points, the 5 points that it has the function. Now, regarding this, we already have discussed that a single DNA molecule, this DNA molecule wraps over the histone proteins. You just remember the last class where we have discussed how the histone proteins form, or the histone cores are formed. The histone core are formed by H2A, H2B, H3 and H4 histone proteins. So, a single core is being formed by these 4 proteins, and this they occur in dimeric structure, and the DNA molecules it wraps over here, take a 1.6 turn, then again it goes back like this, and in this way now it forms the packaging, and each such bits they are called as what? Nucleosomes. We already have discussed it. So, this nucleosomes condenses into a chromatin fiber. So, this is the structure of a chromatin fiber, and it further condenses, the chromatin fiber condenses further, giving rise to this chromatin forms of a chromosome. So, these are the 2 chromatids, and in this way the chromosome is being formed. And again this is another picture where the DNA molecules, now this is a better picture. Now, here the core of 8 histone molecules, now it wraps, this gives you a clear picture that how it wraps and it binds to the histone proteins, and this is again another histone protein that is called as histone H1. Okay, and this histone H1, these are the histone H1, they are they bind to the linker DNA. Now, here you can see these are the linker DNA. That means when 2 nucleosomes they are connected to each other by a gap which is known as the linker DNA. Now, this further condenses. Now, here the nucleosome packs into a coil that twists into another larger coil. Okay, and this supercoiled structure is called as chromatin fibers. Now, in this way, now here the chromosome is formed, and you can visualize the picture or a plate like this at which phase? At the mitotic cell or at any cell division. Okay. Now, this is a clear picture how the DNA molecule wrapped over the histone protein or histone octamer, and how it forms the chromatin fiber, and finally it forms the chromosome structure. Now what is telomere? Telomere is the end of the chromosome. Okay. Now if we have a structure like this, now if this is a chromosome, now these are the telomeric ends. So these ends, these are the telomeric ends, that means the end of the DNA or the end of the chromosome. Telomeres are made up of repetitive sequences. The telomeres, they are made up of repetitive sequences and they are of non-coding DNA. That means here the DNA does not code for any protein. So these are repetitive sequence and generally the telomeric ends does not have any gene component. Okay. So each time a cell divides, this is very important. Each time a cell divides, the telomeric end, it becomes lesser, okay, and it becomes shorter or lesser, and eventually the telomeres become so short that further cell division cannot happen. Okay. Once again I am reading, each time a cell divides, the telomeres become shorter. Eventually the telomeres become so short that the cell can no longer divide. That means division after division, the telomeres, they go shorter, 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 and further no division can occur. Okay, so let us have a look at the diagram. Now these are all, these yellow portions, okay, these all yellow portions, these are all the telomeric end of any chromosome, okay, and obviously you will find them within the nucleus. Now this is the clear cut picture. Now these are the telomeric end, okay, and they are made up of they are made up of repetitive sequences, okay. So they are made up of repetitive sequences. Now here you can see this is something like GGTT, GG, GGTT like this. So 4G, 2T. 4G, 2T. So these are all repetitive sequences. Now here chromosome of an adult cell, now it shortens after multiple replication. So like this it shortens, okay, and finally telomeres at senescence stage, now you will find almost no telomeres or very short telomere. Now it cannot divide further, okay. Now the chromosome of an immortal cell, now it always remain intact, okay, it always remain intact by an enzyme known as telomerase, okay. Now this is the telomeric end. Now again this is another electron micrographic structure of a metaphasic chromosome. Now this is a better picture. Now let me write it here. This is the complete structure of a chromosome. Okay. Now let us have a look what are the features it has. Now we already have discussed there is a central point mostly which are called as centromere. Now two chromatids are there, they are joined at the position of centromere. Then the end portion of the chromatids, they are called as telomeres, and as per the position of the centromere we may have a short arm and a long arm. This is the simple structure of a chromosome, but sometimes in some special kind of chromosome, now we may have some different kind of structures or some more or some extra structure. Now what are these? Let us look. Now telomere we know. Now after that, what are the different things that we have is that now centromere we have previously, this is the typical structure. Telomere is also. Now these are the two chromatids. Okay. Now what are the extra structure? Now you will have a look. Now there is a primary constriction. Now that means now within this chromosome, now at some point, now there is a constriction like this. Okay, and this is what? This is called as primary constriction. And the primary constriction, it is always associated, or this is very near to the centromeric region. This is very important. So, the primary constriction, it is always near to the centromere. Okay. Now, there is a very, you can see it here, there is a portion here which is known as kinetochore. Okay, this is known as kinetochore, and this kinetochore are for what? This is for attachment of spindle fibers. Previously, while we were discussing centromere, before some slides, here you can see that yes, during mitosis, spindle fibers attach to the centromere via kinetochore. So, let us have a look that what are kinetochores. Now, here this is the kinetochore to which the spindle fibers, okay, all the spindle fibers, they attaches itself to this kinetochore. Okay. Now, there is again another constriction, okay, there is again another constriction which is known as secondary constriction, and this is what? This is the nucleolar organizer. So, this is the secondary constriction known as nucleolar organizer. As I told you that now you know that, let us suppose have a discussion in mitosis. Now, the mitosis has 4 phases. The first phase is known as, or if we can go into more details, now a cell cycle has an interphase and a dividing phase. Now, the interphase comprises of G1 phase, S phase and G2 phase. G1 phase is the phase where lots of protein and RNA synthesis occurs, and it prepares itself for the replication to happen. For replication means for DNA synthesis to be happen during the S phase. Now, next phase is the S phase. Now, the S phase is the synthetic phase. Now, what is being synthesized here? Here the DNA is being synthesized, or you can say the phase as DNA replicative phase, DNA duplicative phase, or DNA replication or DNA duplication. So, the DNA is synthesized here and the DNA content is doubled during the S phase, and it happens once per a single cell cycle. So, after S phase, now the G2 phase, it prepares, or lots of protein and RNAs are synthesized, and here it prepares itself for the dividing phase. Now, the dividing phase comprises of 4 phases. What are those? Now, these are like what? Prophase, or this is what known as. So, the dividing phase has again 2 phases. One is kinetokinesis, or your kinetokinesis or cytokinesis. So, the division of nucleus happens, nuclear division, it happens in 4 phases. One is prophase, the other one is metaphase, third one is anaphase, and the fourth one is what? Telophase. Okay. Now, during prophase, lots of things happens. Now, what happens? Now, here at the commencement of this nuclear division, the prophase, okay. Now, the nuclear membrane and the nucleolus, this disappears towards the late phase of prophase, no doubt, but slowly the nuclear membrane and the nucleolus, it disorganizes, or it generally disappears, okay. And the chromosomes which are not being visible during interphase, slowly they are visible in the form of small threads, okay, having a single dot somewhere in between, and this is called a centromere. So, the chromosomes appears in the form of thread, slowly it condenses into that metaphasic structure of chromosomes showing two sister chromatids, okay, and the chromatin fibers are much condensed there, okay. So, what happens now, the nuclear membrane and the nucleolus, it completely disappears. Now, after the metaphase and anaphase, now when comes the telophase, now during telophase, right, this is a cell, the set of chromosomes, now they would have reached to the two poles, okay, towards the two opposite poles of the cell being held by the spindle fibers, okay. Now, before the cytokinesis, there is reappearance of nucleus forming the nuclear membrane. Now, here during telophase you can find two daughter nuclei within a single cell, okay. So, what happens here? Now, how this nuclear membrane, it is formed, and again the nucleolus, it again reappears. How it is being formed? So, the nucleolus reappearance, it happens because of what? The secondary constriction. That means, this is the reason where the nucleolus, it reorganizes, okay. So, that is why in short also this is called as nucleolar organizer, or in short this is called as NOR, N-O-R, NOR, nucleolar organizer, okay. So, this happens during secondary constriction, or it happens at the point of secondary constriction. So, these are the points of secondary constriction and these are the points of primary constriction, okay. So, primary constriction and centromere, they are very much close to each other. So, there is no demarcation between these two also sometimes. So, and all the chromosomes, they do not have the secondary constriction. Very few chromosomes, they would have the secondary constriction, at which the nucleolus reorganizes itself at the telophase stage of the mitotic or any kind of division, or the meiotic division or whatever. Now, during the telophase, the reappearance of nucleolus happens at the secondary constriction. Now, because of presence of secondary constriction, what happens? Again we get another kind of structure within the chromosome, that is called as what? Satellite, okay. So, satellite is again another structure of a chromosome, now which generally it forms, it is being formed by, or it appears as a protuberance, or it appears as a short tail, or whatever you can say. So, it appears as a short end, okay, because of the constriction happened by secondary constriction, okay. So, one is a primary constriction, close to it there is presence of kinetochore, where the spindle fibers get attached, or it attaches itself to the spindle fiber. So, one at this place, another at this place, and another reason is that is secondary constriction, where the what? Nucleolar, or this is called as nucleolar organizer, or where the nucleolus reorganizes itself at these points. And because of the presence of secondary constriction, we have a small satellite like portion, or we have a small portion which is being cut out from the main portion of the chromosome, and it appears as a short tail or something like that, and we call it as satellite, okay. So, what we call it as? We call it as satellite. Okay. Okay, anyways, now this is the structure of a chromosome once again. Now, what are the main features? Now, there is a centromere, there is telomere, okay, and there are the chromatids, there is a short arm and a long arm. Well, besides that, some of the chromosomes, they have a primary constriction near to which there is a kinetochore, okay, and there is a secondary constriction which gives rise to a kind of structure known as satellite. Well, so we will go to the next one. Now, we will classify the chromosome. After considering the structure of chromosome, we will classify the chromosomes. Well, so on what basis we will classify chromosomes? Some chromosomes will be short, some chromosomes will be long, okay. As per the length we can classify them, as per the position of centromere we can classify them, as per the numbers of centromere we can classify them. So, various bases are there by which we can classify the chromosomes. Now, what are the bases? On the basis of the type of genes. Well, so on the basis of the type of genes we can classify the chromosome once as autosomes, the other one as allosomes, or in other words they are called as idiochromosomes or sex chromosome. This very much simple term is sex chromosome. So, the autosomes or allosomes. So, on the basis of genes what we can classify them? Autosomes or allosomes. Let us have an example. Now, in human being, now what is the 2n? This is equal to 46. Now, how many number of pairs we have? We have 23 pairs. We have 23 pairs of chromosomes. Now, from out of this 46, we have 44 numbers of allosomes or 22 pairs of chromosomes in human beings. Now, this is for human beings. Okay. So, in human beings, now 22 pairs or 44 numbers of chromosomes are autosomes, whereas 2 numbers of chromosomes are allosomes, or it forms a single pair of sex chromosome. Okay. So, what are the sex chromosomes? One is X, the other one is Y. So, X and Y chromosomes are the sex chromosome, as we know everyone. So, on the basis of genes, we have either autosomes or allosomes. Now, next, on the basis of the number of centromere. Now, if we have a single centromere, now what we will call the chromosome as? If we will have 2 numbers of centromere, then what we will call it as? If we have more numbers of centromere, what we will call it as? And if we do not have centromere, then what we will call it as? So, on the basis of number of centromere, it may be monocentric. Monocentric means when there is a single chromosome, when there is a single centromere. Okay. So, when the centromere is 1, we will call it as monocentric. Okay. When the centromeres are 2, we will call it as what? Dicentric. The example is, we can find it in males. Some of the chromosomes, they are dicentric. Now, when it would be more than 2, 3, 4, 5 or something. Now, when the centromeres are more than 2, we will call it as polycentric. Okay. And finally, if we do not have any centromere on a chromosome, then we will call it as what? Acentric. Okay. So, on the basis of genes, it is autosome and allosome. On the basis of the number of centromere, now it is called as monocentric, dicentric, polycentric and acentric. Now, the most important that we generally consider the basis of classifying the chromosomes are the position of the centromere. We can classify the chromosomes in various ways, but this basis is the most important one. We generally consider this one. and this basis is the position of centromere. Okay. So, on the basis of the position of centromere, now we can classify them as metacentric, subcentric, acrocentric and telocentric. Okay. So, on this basis we can call it as meta, submeta, acro or telocentric. Let us have a look on this one with the help of a diagram. Now, this is the classification of chromosomes based on the position of centromere. So, basing upon what? The basis is the position of centromere. Let me explain first. Now, if this is the chromosome, okay. Now, let us suppose the centromere is present at one terminus, then what we will call it as? If it is present somewhat below that, now what we will call it as? If it is present somewhat below that or at the central point, now what we will call it as? Like this, okay. Now, on this basis, now here you can see the telocentric chromosomes. Now, the telocentric chromosomes are what kind of chromosomes? Where the centromere is present at one terminus, okay. That means where there should be telomere, we have centromere, okay. So, the telomeric end and centromeric end has no distinction or they have no distinction, okay. So, at that point, now when the centromere is positioned at a location of telomere, it is called as telocentric chromosomes, okay. Now, acrocentric means when it is very much near to the telomeres, that means when the centromere is very much near to the telomeres. So, if this is the telomere, the centromere is more or less close to the telomeres, but not exactly at the telomeric region, then we will call it as acrocentric chromosome, okay. Now, here we have, in case of acrocentric chromosome, generally we have the satellite, the P arm and of course the centromere and the Q arm. P arm means the upper portion or the short arm is the P arm and the lowermost one is the Q arm, or just above the centromere it is P, below it this is Q. So, next, now when the centromere is located at a position, okay, when it is located at a position more or less or very near to the central portion, not exactly at the central region of the chromosome, rather somewhat above or below the central region, then we will call it as what? Submetacentric. That means metacentric, submetacentric. Metacentric means when the centromere is positioned exactly at the middle point, okay. So, it is not exactly the middle, but it is just above the middle or below the middle, okay. So, this is called as submetacentric. Here this is the centromere, this one is the P arm and this one is the Q arm, okay. Next, the metacentric chromosome. Now, here when the chromosome or the centromere is positioned very much at the middle of the chromosome, now it will call it as metacentric chromosome, okay. So, here this is the P arm, this is the Q arm, and the P and Q arms, they are what? Equal in size or the distance or the length of P arm and the Q arm, they are more or less the same, or they are, let us suppose the P arm is 0.1 nanometer. So, Q arm will be also 0.1 nanometer like this. Well, so the length of both the arms P and Q, they are equal to each other. Well, so once again, on the basis of centromere, on the basis of the location of the centromere, now we can call it as telocentric, acrocentric, submetacentric or metacentric, okay. Now, when the centromere is at the telomeric end, it will be called as telocentric. When the centromere is very much near to the what? when it is very much near to the telomeric region like this, then we will call it as acrocentric, and here we will find a satellite, okay. And of course this is the P arm and this is the Q arm. P is the short, Q is the long arm. Then submetacentric means when the centromere is very much near to the middle position, then it will called as submetacentric, and when it is exactly located at the middle position of the chromosome, okay, then it will be called it as metacentric chromosome. So what we have discussed? We have discussed the structure of chromosome, the typical structure of chromosome that comprises of a centromere, two chromatids, the telomeric ends, accordingly the short arm and the long arm. But some specialized chromosomes, they have some extra structures like the primary constriction, to which the kinetochores are present, to which the spindle fibers will attach themselves at the time of cell division, and there is a secondary constriction otherwise known as nucleolar organizer, or in short NOR. This is the location at which the nucleolus reappears, or it reorganizes itself at the time of telophase or at the time of cell division of the telophasic phase. So, this secondary constriction gives rise to a kind of structure known as satellite, and these are the typical, or these are the total structure of chromosomes, of various chromosomes. Now, apart from that, on the basis of several facts, we can divide the chromosomes into different types. So, on the basis of gene, we can divide it into autosomes and allosomes. On the basis of the numbers of centromere, we can divide it into like monocentric, dicentric, polycentric and acrocentric. And finally, on the basis of the position of centromere, we can divide it into telocentric, acrocentric, submetacentric and metacentric. Okay. So, this is regarding structure of chromosome, then classification of chromosomes. Next, we will go to another topic, another important topic. What is that? Let me write it here. Now, we will discuss today heterochromatin okay, heterochromatin and what? Euchromatin. So, this is another important this is another important part of chromosome, that in case of heterochromatin, the chromatins are hetero in nature. That means it takes a very dark stain. I will tell you why. So, it takes a very dark stain, and here it takes a very light stain. Okay. So, what is the importance of heterochromatin and euchromatin? Let us have a look. Okay. Now, what is heterochromatin and what are euchromatin? Now, we will discuss this too on basis of various parameters, or we will compare on several bases like definitions, staining, DNA conformation and etc. Okay. So, what happens now? Next, let us have a look on the definition of heterochromatin and euchromatin. Now, heterochromatin is a tightly packed or condensed DNA that is characterized by intense stains when stained with nuclear stains and transcriptionally inactive sequences. Okay. So, it takes a very dark stain or intense stain. So, as I told you that it takes a very dark stain. Now, if you would be confused, now let me give you just a diagram. We will discuss it at the last. Now, you see, now which one is the heterochromatin region? Now, you see which would have taken a dark stain. Now, you can see there are some light stains. Now, these are all chromatin fibers. Okay. So, these are all chromatin fibers. Some of the portions or some of the reason they are taking the dark stains, some of the reason they are taking light stains. So, the darker stain regions are called as heterochromatin region because they are packed with repetitive sequences and they are very much the DNA is very much tightly packed here. So, it is taking much stains. So, here the color is intense or darker as compared to the euchromatin region. So, let us discuss the comparison between the heterochromatin and the euchromatin. So, what are heterochromatins? These are more condensed and here the silence genes are there. The gene is very poor, it is highly rich in AT content and the stains are darker. As you can see here, here the stains are darker. So, these reasons, this is also again another reason. So, the red colored portions that I have marked here. So, these are the heterochromatin regions. The rest of the regions are euchromatin because you can identify the coloration difference. Okay. So, the intensity is high here and the intensity is lower at the lighter states. So, the euchromatins are less condensed, they are highly they are high in gene expressions, they are gene rich and the stains lighter color. Okay. So, let us have a comparison between the heterochromatin and the euchromatin. Now, the euchromatin is a more lightly packed DNA that is characterized by less intense staining and DNA sequences that are transcriptionally very active and might become transcriptionally active at some point during growth. Either they are transcriptionally active or they will be transcriptionally active at any point of the growth. Okay. Now, what kind of, so this is regarding the definition, this is regarding the definition. Now, next, what kind of stain they do take. Now, you imagine both of the portions, they are present on the same chromosome. So, there is a single chromosome, some portions are very intense stain or they are darkly stained and some portions are lightly stained. So, the darker stain portions, they are the heterochromatins and the lighter strain portion, they are the euchromatin region. So, regarding staining, the darker strain are heterochromatins, the lighter strains are the euchromatins. Now, regarding DNA conformation, now the DNA is tightly bound here or condensed. This is very much condensed or tightly bound, whereas in euchromatin, this is lightly bound or they are very much compressed. Here they are very condensed, here they are just compressed. Okay. So, they are just lightly bound because why? Because these are the regions which are transcriptionally active. So, in order to have transcriptionally active DNA region or transcriptionally active chromosome region, here the DNA has to be exposed. Well, so that is why they are very lightly packed there. So, next, the DNA in heterochromatin is folded with histone proteins. Obviously, as the packing is very much condensed, so they are heavily folded with histone proteins. But in euchromatin, this kind of folding is not seen, rather it is unfolded to form a beaded structure. Okay. So, in euchromatin, now this histone proteins would be what unfolded or the DNA is unfolded to get exposed in order to undergo the process of transcription. So, they form a beaded structure. So, this is regarding definition, staining and DNA conformation. Now, next regarding genes. Now, the heterochromatins, they are high, they do not have genes or they have very few genes or almost they do not have any genes. Okay. So, the genes present in these regions are usually inactive. So, there is no such activity. Now, the genes present in euchromatin is either already active or they will be active during some point of time. Okay. Now, regarding transcription, obviously, whenever there is more gene expression, there will be more transcription. So, heterochromatin region will be transcriptionally inactive and the euchromatin region they will be transcriptionally active. So, as you can see it here, this is transcriptionally inactive and this is transcriptionally active. Now, regarding the DNA content, the Heterochromatin has more amount of DNA. Obviously, as it is very tightly packed, it is tightly compressed or condensed. Obviously, there would be more and more amount of DNA would be there. So, the DNA content is more, or this more amount of DNA tightly compressed with the histone proteins, and we already have discussed this. Sorry. So, euchromatin has less amount of DNA lightly compressed with the histone proteins. Why so? Because in order to undergo the active process of transcription, now they have to be what? They have to be lightly bound with the histone proteins. Well, now the content in genome, heterochromatin forms a very smaller part of the genome. In humans, it is about 8 to 10% of the genome. Okay. Now, whereas in euchromatin, now it forms 90 to 92% of genome. So, it forms a more significant part of the genome. So, what happens? So, what happens here? Content of genome is low here and it is high here. Now, the heterochromatins are found in eukaryotes only, whereas the euchromatin, they are found both in prokaryotes and eukaryotes. Now, the types are, it exists in two types, one is constitutive, the other one is facultative, but here it is only constitutive. Location with the nucleus, it is present towards periphery, whereas else centrally or towards the inner body of the nucleus. They are centrally located or towards the inner body of the nucleus. Now, it exhibit heteropyknosis, but it does not exhibit heteropyknosis. Now, replication, they replicate later to the euchromatin, but euchromatin replicates faster than heterochromatin. Okay. So, it is a late replicative portion and these are the early replicative portion. Now, genetic process, heterochromatin is not affected by genetic process, but it is affected by, the euchromatins are affected by genetic process. And finally, what is the function? It maintains the structural integrity of the genome. Heterochromatin maintains the structural integrity, whereas it allows the genes to be transcribed and variation to occur within the genes. Okay? So, the function is that it maintains the structural integrity, it is functional in nature. That means, here the genes are actively transcribed and variation occurs within the genes. And finally, what are the portions where you will find the heterochromatin region? This is mostly towards the telomeric region and towards the centromeric region. So, here you can find at telomeres and centromeres, whereas besides that you will find the heterochromatin in rest of the portion of the chromosome. So, lastly, let us have a look once again. So, where you will find the heterochromatin? At the telomeric region. So, here these are the telomeric region, at the centromeric region, now near the centromere, okay, and again at the telomeres. So, these are the regions of what? Heterochromatin, and this is, and this light colored portion, these portions, these are the portions of the what? Euchromatin. So, these are the concept of the structure of chromosome, the classification of chromosome and the kind of chromatin on the basis of the kind of stain that it takes. This is all about the chromosome. So, this is all about the structure of chromosome, and here we finishes the structure of chromosome. In the next class, we will go for a new topic. Thank you. Have a good day. चाहिए